Now that we've looked at glycolysis and gluconeogenesis, I want to look at the citric acid cycle because the pyruvate, the product of glycolysis, can then be further oxidized so that its carbons can enter the citric acid cycle. To be clear, other terms which mean exactly the same thing as a citric acid cycle is Krebs cycle, tricarboxylic acid cycle, and TCA. Tricarboxylic acid because a number of the intermediates in the citric acid cycle have three carboxylic acid groups on them. Just like with glycolysis and gluconeogenesis, I don't want you to memorize a single enzyme name, but I want you to be able to interpret every one of those enzyme names. In particular, you should be able to identify the substrate in the product based on the name, maybe have some idea of how it fits into Krebs cycle. Also, a number of the steps in Krebs cycle involve oxidation driving a reduction, so you should be able to know what substrate or product is oxidized, which one is reduced, which substrate undergoes an oxidation or a reduction, what goes up in energy, what goes down in energy. Again, comparing this to the glycolysis and gluconeogenesis talks, we mostly looked at the enzymes which connect glycolysis to the pentose phosphate pathway, or to gluconeogenesis, or to the pentose phosphate pathway. So in looking at the citric acid cycle, I would like to look at what connects glycolysis to the citric acid cycle, and the focus is going to be on pyruvate and on acetyl coenzyme A. Will you list the compounds which can be formed out of pyruvate through a single chemical reaction, and for each of them list the number of carbon atoms? It turns out the number of carbon atoms is going to help you sort things out. In fact, the information I'm looking for is right here in the figure, so go ahead and fill it out, but I'm going to move on. Pyruvate can be converted to alanine with a simple reaction. Pyruvate can be used to make acetyl coenzyme A. Pyruvate can be converted to lactate, and pyruvate can be converted into oxaloacetate. Pyruvate can be converted to alanine. Now, can you tell me what type of reaction? Pyruvate can be converted to alanine by exchanging the ketone for an amine to make an amino acid instead of a keto acid. Or alanine can likewise be converted into pyruvate by a deamination or a transaminase reaction, exchanging between the amine and a ketone. Or pyruvate can be converted to lactate. Do you know what enzyme does that? We've looked at it a lot. How about lactate dehydrogenase? And again, that tells you about the energetics and about what coenzyme you think should be used. And then, of course, pyruvate can be converted to oxaloacetate, or pyruvate can be used to make acetyl coenzyme A. Why do you think I colored two of these arrows red and two of them green? Please pause and answer. Two of these reactions are reversible, as indicated in green, with arrows on both sides of the line, and two of these reactions are irreversible. There's a drop in energy so that they cannot go back uphill, and that's indicated with the red arrows. Can you name each of the four enzymes responsible for these reactions? Please go ahead and do so. And for that matter, will you also list possible uses for each of these products? Please go ahead and do so. It's in your interest to do this right now, and I think you can simply by interpreting enzyme names. Well, of course, if you convert pyruvate to alanine, well, you have an amino acid. You could use it to make protein. Lactate dehydrogenase allows the interconversion between lactate and pyruvate, and this is really useful in sharing metabolism between cells. For example, your red blood cells are exclusively anaerobic and make a lot of lactate, which they release into the blood. Then the lactate can be filtered out by the liver, converted back into pyruvate, and sent into gluconeogenesis so that the red blood cell can share its anaerobic metabolism with the aerobic metabolism of the liver. It's real important to know the difference between lactate dehydrogenase, pyruvate dehydrogenase, and pyruvate carboxylase. The names tell you exactly what they do. An important distinction is that pyruvate carboxylase is the first step of gluconeogenesis. So that's important. It's also important to know that pyruvate carboxylase is also used to replenish citric acid cycle intermediates. So let's say that you're really low on sugar, your brain needs sugar, and you're running a whole lot of gluconeogenesis in the liver while you're converting oxaloacetate into sugar. And that can deplete the intermediates in the citric acid cycle. In order to replace them, you would want to use pyruvate carboxylase, which takes pyruvate, which is not a citric acid cycle intermediate, converts it to the 4-carbon oxaloacetate, which is now a new citric acid cycle intermediate. Let's compare pyruvate carboxylase to pyruvate dehydrogenase. Pyruvate dehydrogenase is used to make acetyl coenzyme A and carbon dioxide. Then acetyl coenzyme A can donate its 2-carbon acetyl group to oxaloacetate to make citrate. So it has increased the number of carbon atoms from 4 in oxaloacetate to 6 in citrate, and now you can burn it. But because you are simply converting one citric acid cycle intermediate into a different citric acid cycle intermediate, you have not changed the net number of intermediates. So that is, pyruvate dehydrogenase cannot be used to replenish the number of citric acid cycle intermediates. If you're low on citric acid cycle intermediates, you need to use pyruvate carboxylase, or you might be able to convert some amino acids into that also. In addition, this pyruvate carboxylase, which takes a 3-carbon pyruvate and makes a 4-carbon oxalic acid, is important enough that I don't just want to point out the reaction and give you the name of the enzyme, but I want to actually show the reaction because I think seeing it visually will help you remember it.
Pyruvate carboxylase, that tells you it adds a carbon atom, and that is going to take pyruvate and convert it into a citric acid cycle intermediate, which means the number of citric acid cycle intermediates has increased. In contrast, pyruvate dehydrogenase takes 2 carbon atoms and links them to oxaloacetate to make citrate. So it has not increased the net number. If you're deficient in citric acid cycle intermediates, and if you want to increase them, you want to use pyruvate carboxylase. If you simply want to make ATP with the citric acid cycle, you would like to use pyruvate dehydrogenase. Those are not the only enzymes that can do that. For example, you can look at fatty acid oxidation can also make acetyl coenzyme A impede the citric acid cycle. But my question is, how will you determine whether pyruvate dehydrogenase is going to fuel the citric acid cycle, or whether you want pyruvate to be converted into a citric acid cycle intermediate, which could either replace or replenish, increase the number of citric acid cycle intermediates, or just directly fuel gluconeogenesis. It's just like the control enzymes between the 3 different pathways for hexosphosphates. Now we're looking at how you control between making more intermediates and just running the citric acid cycle.
Oh, sorry. I had a quick question.
Yes.
I was just wondering, I thought pyruvate carboxylase made oxaloacetate.
It does.
Okay, so then that for pyruvate carboxylase, that wouldn't change the number of intermediates, correct? Because you're just making directly.
No. Pyruvate carboxylase absolutely changes the number of intermediates. You take pyruvate, you add carbon dioxide to it, and make oxaloacetate. Pyruvate is not a citric acid cycle intermediate. There are 8 intermediates, There are 8 intermediates and pyruvate is not one of those. So in
Oh,
other words, if you take pyruvate and oxidize it to make acetyl coenzyme A, acetyl coenzyme A can feed the citric acid cycle, but that's by converting oxaloacetate to citrate. That does not, that contains one citric acid cycle intermediate and makes another. It just feeds and runs the cycle. But let's say you've been running gluconeogenesis because you're starving and you have a very few numbers of each citric acid cycle intermediate left. Well, you can run it, but you're going to have trouble running it on so few intermediates. You need more intermediates. And the way you can do that is by taking something that is not a citric acid cycle intermediate, adding a carbon in making a citric acid cycle intermediate.
okay, gotcha. Thank you, Dr. Hansen.
It's a real important question that confuses a lot of people, including myself sometimes, okay? So, and I'm really glad that we slowed down on that because that's a really foundational thing. Now, I have a question. Do you think that ATP is going to speed up the rate of the citric acid cycle or slow it down? The citric acid cycle is regulated by ATP.
Slow it down.
That's really good. Now, who's that? Is that you again, Brent?
That was me, Dr. Hansen. My name is Vartan.
Barton, good answer. And why do you think ATP will slow it down?
Because we know that glycolysis is driven to, or not glycolysis, cellular respiration is driven to make energy. And if we already have a lot of high energy compounds in our body, then we would want to slow down making so much energy.
You are exactly right. Just as ATP downregulates glycolysis, if you have a real high concentration of ATP, you don't want to do glycolysis. You don't want to do the citric acid cycle. So it tends to slow it down. And AMP?
AMP would be the opposite. AMP is a low energy coenzyme, so it would drive cellular respiration, glycolysis.
And this is exactly what happens, and it actually happens at several steps in the citric acid cycle with ATP and AMP. Now, for someone else, how about NADH? If you have a real high concentration of NADH, will that speed up or slow down the citric acid cycle?
Having a lot of NADH would mean that we would slow down this TCA cycle.
Exactly right. Yeah. So we're going to move on now, and we've already talked about the mechanism of pyruvate carboxylase, so I'm not going to look at that more. But I will tell you that also it turns out that acetyl coenzyme A activates pyruvate carboxylase. Why would it be doing that? In fact, if acetyl coenzyme A is building up, what does that suggest about the citric acid cycle and glycolysis?
Would it suggest that we have low amounts of intermediates since we cannot combine it with oxaloacetate to form citrate?
That's exactly right. If that one way you could have a buildup of acetyl coenzyme A is if you don't have enough citric acid cycle intermediates, so the glycolysis is going at a faster rate. Another way that could happen is that say you're starving and you need to run gluconeogenesis. Well, that can be fed by fatty acids. So you can get a huge buildup of acetyl coenzyme A being fed into the citric acid cycle. In the buildup of that can say, wait a minute, if we have pyruvate, we don't want to just burn it. We need sugar. So let's activate pyruvate carboxylase and let's downregulate pyruvate dehydrogenase. If you may be getting tired of seeing lactate dehydrogenase, the reason I like to talk about it is because this reaction explains a lot of the citric acid cycle and a lot of oxidative phosphorylation. And I found that some people are still having difficulty with it. So bear with me if you listen to the pre-matriculation and you say, okay, it's simple. I don't think that the majority of you feel that way, but I'm sure some of you do. But the point is that all enzymes work by bringing a coenzyme next to a substrate, and they position them right where you can exchange something. Lactate dehydrogenase grabs lactate and NAD+, and it puts that hydrogen atom right next to the aromatic ring, and it pushes them together so that the hydride can move over. Now, and in fact, I recommend that if you're, say, I already know this, go ahead and draw the arrows to show how that can happen. And the thing is that what's going to happen is that the electrons on the hydroxyl can resonate down to form a double bond and push the electrons in the double bond onto the nicotine amide. So now you have a high energy nicotine amide. And again, this is in the preview video. I want to say that if you didn't get it then and don't get it now, please contact me to be sure you understand it before we get into oxidative phosphorylation next week. Because this is going to be over and over that we're pushing these high energy hydrogen atoms and this reaction can run either direction. Since it is called lactate dehydrogenase, that grammar tells you that the forward reaction is acting on lactate and it's removing a hydrogen from lactate so that you end up pyruvate having one more bond to oxygen. The reverse reaction of lactate dehydrogenase takes the hydride off of the nicotinamide and it nucleophilically attacks pyruvate, the electrons resonate up, and you end up with lactate. That's just based on enzyme nomenclature, what direction you're going. It turns out, as I mentioned earlier, that since the hydride on lactate is high in energy and the hydride on NADH is high in energy, that the relative energy of lactate plus pyruvate is almost the same as the relative energy of NADH plus NAD plus, so that this is, it's like an isomerase almost. It's not because but because one substrate has gone up and one has gone down in energy. But the reason I wanted to say that is that the pyruvate dehydrogenase does exactly the same thing, but it does it in multiple steps and in a more complicated way. And again, we just talked about the regulation of this enzyme. So this would be a good practice thing sometime on your own after the class. When you look at this thing, coenzyme A, think adenine, or think A-adenosine, adenine ribose phosphate with a linker arm, which is basically pantothenic acid, an acid that is found everywhere. Panto means everywhere, ate means an acid. Anywhere you have oxidative metabolism, you're going to have pantothenic acid, that vitamin linked to ADP to give it this long extender arm. And the business end is a simple sulfhydryl or an acetyl group linked to the sulfur. Now, can you tell me which is high energy and which is low? Is a sulfhydryl a high or an acetyl high?
I would think the sulfhydryl is the higher energy here.
Well, you know, I like your answer because you're saying, wow, that's a hydride ion, right? You're saying that's a reduced hydrogen. And I want to point you that unfortunately, that's only a pair and I have a full house. The acetyl group actually has 3 hydrogen atoms on it. Do you see that? And you are correct, the sulfhydryl is intermediate in energy because that sulfhydryl can get oxidized to a disulfide. But between these two, you have to look at the whole thing. Here we have only a hydride that can be driven somewhere, whereas here we have an entire acetyl group which has 3 bonds, 3 carbon to hydrogen bonds and 1 carbon to carbon bond. So it's a little bit of a trick question, but I get confused on that too, that you can't just say a sulfhydryl is extremely low, but it is certainly lower than the acetyl group. So I like your answer, thank you. But now we're looking at the entire citric acid cycle, and I'm hoping that all of you noticed that there's one foundational activity of the citric acid cycle, a learning objective, and that is what? Why is it that of the 8 enzymes, 4 of them are the same type. And what is that type? Can someone tell me?
Looks like we have a lot of dehydrogenase going on.
Yes, we have a lot of dehydrogenases here. Look at that. This cycle, you know, whenever anyone teaches you the citric acid cycle, the first thing they should say is, look at this. This thing has a motive. I am suspicious of this guy's politics. I think he is only interested in making one particular compound. And what would that be?
Carbon
NADH.
NADH. And this is the citric acid cycle. It is one NADH making machine. Now, I have to point out, I'm exaggerating, right? You got me on that. It does take one FAD and convert it to FADH2. But again, remember that when you're talking about dehydrogenases, most of those reactions, the most energetic ones, take NAD plus and reduce it to NADH. But you can also take FAD and convert it to FADH2. And what's real interesting about succinate dehydrogenase is that that is not only an enzyme in the citric acid cycle, but the FADH2 in the same enzyme complex can directly dump the hydrogen atoms with its electrons into the electron transport chain. So if you really do want to know these and the dehydrogenases in the citric acid cycle, and they're all important. Isocitrate dehydrogenase, alpha keto glutarate dehydrogenase, both of those are negative energy steps. They plummet in energy, and ATP and AMP regulate them to drive the cycle. But succinate dehydrogenase is important because it is involved with the citric acid cycle as well. And now you also have pyruvate dehydrogenase. You know, you pump that pyruvate into the matrix of the mitochondria, you're going to get a lot of NADH. Oh, and I've already answered my question, sorry. What happens to those high energy electrons on NADH in the mitochondrial matrix?
Aren't they going to get oxidized later in the oxidative phosphorylation ETC transport chain?
Exactly. There's a reason the citric acid cycle is making all that NADH in the matrix, because that NADH in high concentration will bump into complex 1 of the electron transport chain, dump its electrons into the electron transport chain to be oxidized back to NAD+, and then it'll go back to the citric acid cycle. You make a bunch of NADH, and that is one of the learning objectives. The citric acid cycle itself, boy, I don't see it here. The citric acid cycle itself makes one GTP. It makes one high energy phosphate. I need to add that to the figure here. But
Your Hansen, it's right there.
Where is it?
Between succinyl CoA and succinate, it says.
Oh, I do show it. You're right. Okay, good. Yeah. Okay. So it's pretty piddly when it comes to making triphosphates, right? I would say lame, but everyone specializes in something. And the purpose of the citric acid cycle is to make all that NADH. And notice, not NADPH. Can anyone out of a review tell me what makes NADPH?
Glucose 6-phosphate dehydrogenase in the pentose phosphate.
Yes, that's why that's such an important enzyme. Not only does it regulate the pentose phosphate pathway, but that's your body's primary source of NADPH, which is used in biosynthesis. All this NADH is NADH because that is the form of high energy hydrides that are used to make ATP. And whenever I say make energy, I talk about making ATP. Now, this is a
Dr.
figure. Yes.
Sorry, on the previous slide, I had a question. So could we interpret the one-way arrows as moving from a high energy to a low energy?
Okay, yes, to be honest here, I saw in my figure that I was just showing it like a unidirectional thing mostly. And I thought I should show it that, hey, what really is unidirectional are the 3 steps: citrate synthase, isocitrate dehydrogenase, alpha-ketoglutarate, that these dehydrogenases are big plummets in energy, and those are the regulatory steps. Now, I want you to understand the regulation of the citric acid cycle, but I didn't make that a learning objective. It's in your interest to think, oh, you know, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, you're making carbon dioxide. This is low energy. There is enough power there to produce NADH, and it goes down enough that it's one directional, so that ATP and AMP regulate the citric acid cycle largely through either citrate synthase or these two dehydrogenases. And the rest really can flow in either direction, kind of like the middle of glycolysis. So that's a really good and important question. In thinking about the citric acid cycle, do you really want to memorize the structure of succinyl-CoA, succinate, fumarate, malate, oxaloacetate? I, for one, don't want to memorize that. But there is a way of simplifying the citric acid cycle and getting insight into how powerful it is, and a little bit of structural insight, I think, from a big view. So we're going to avoid getting into the weeds here. So what's really big is that step we've been talking about, that you can take 4-carbon oxaloacetate and add 2 carbon atoms to it from an activated acetyl coenzyme A with that activated 2-carbon unit to make citrate. So we go from 4 to 6 carbons. Not hard to know. You know, I'm hoping you're very familiar with an acetyl group. I browbeat all of you with that, with what an acetyl group is back in the pre-matriculation. And it's 2 carbons and an oxygen through some type of ester link, right? Well, the thiol ester bond is so high in energy that it can actually form a carbanion here and add basically the, replace the sulfur with an oxygen and add a acetyl group to here through a carbon-to-carbon bond. So that first step, oxaloacetate to citrate, ugly nomenclature, but again, oxalo is almost like oxalate. And now you add another acetate group and you have citric acid. You've gone from 4 carbons to 6. You know there's a 2-carbon unit being added. Now that is simply isomerase. Can anyone tell me what the relative difference in energy is between citrate and isocitrate?
Shouldn't they be roughly the same? Because
no, roughly. That's anytime you hear isomerase, go, oh, isomer. Nothing's being oxidized or reduced. Citrate and isocitrate are almost indistinguishable in energy, so they can flow back and forth. Now, I mentioned how important the dehydrogenases are. So you can basically drive carbon dioxide off this, use the energy to make NADH. And in that process, you've gone from 6 and 6 carbons down to how many?
5.
Yeah. And the reason I mention this is that alpha-ketoglutarate can be used to make an amino acid. Anyone want to make a wild guess? Is it tryptophan?
No.
Phenylalanine? Go ahead, make my day.
Glutamate, your favorite
yes.
acid.
And what's so neat is you have 8 intermediates. Only a single one has 5 carbons, and it's named glutarate. And we are going to look at this in painful detail during neuromuscular when we look at glutamate as a neurotransmitter, and we're going to look at it during GI when we're looking at glutamate being used to process nitrogen for the urea cycle. And also knowing that alpha-ketoglutarate, the only 5-carbon member, really helpful. So we have 6 and 5, and then alpha-ketoglutarate dehydrogenase. We have these 2 adjacent dehydrogenases. You knock off one CO2 and make NADH. You knock off a second CO2 and make succinyl coenzyme A. And if you look, 4 carbons, 4 carbons, 4 carbons, 4 carbons, 4 carbons, 4. This is all 4 carbons. Now, you could say, wait, this has 4 carbons plus coenzyme A. But I'm saying you have the thiol ester bond. There's a 4-carbon intermediate attached to coenzyme A. It's broken off. and then you simply modify it to get back to oxaloacetate. You will hear about succinate, fumarate, malate, oxaloacetate, and succinyl coenzyme A. They're going to play many important roles. Don't learn their structure, just go, oh, succinate, that's going to be one of the citric acid cycle intermediates. As long as you know they're an intermediate, you know that you're going to have a lot of it in the mitochondrial matrix. So the way of thinking about the citric acid cycle in terms of algorithms is to think of adding a acetyl group to go from 4 to 6, and then knocking one off, the next off, you're back to 4 carbons, and you've gone through these important intermediates. Just as alpha-ketoglutarate can be used to make an amino acid, also oxaloacetate in a single step can be made to make an amino acid. And can someone tell me what that amino acid would be?
Aspartate.
Yeah, and in fact, if you look at this, right, it's a single step. We have 2 carboxylic acids on it. If we're going to make an amino acid, we're going to replace the ketone, not one of the carboxylic acids, but the ketone with an amine. Just as in alpha-ketoglutarate, we replace the ketone with an amine. Here we have a 5-carbon intermediate making glutamate. Here we have a 4-carbon intermediate being converted to aspartate. Say you need to do some protein synthesis and you have plenty of energy, but you don't have enough glutamate, or you need a neurotransmitter glutamate. You can take that alpha-ketoglutarate, transaminase it, and you're up and running. You can take oxaloacetate, make aspartate when you need it. These 2 reactions are extremely important in nitrogen metabolism and in neurotransmitters. And oh, I guess I showed you this one already, but it is in your interest to think, if I need amino acids, I can take pyruvate and make alanine. If I need keto acids, I can take alanine and make pyruvate. Likewise, if I need amino acids, I can make aspartate from oxaloacetate or oxaloacetate from aspartate. And likewise, alpha-ketoglutarate can be used to make glutamate, or glutamate can be used to make alpha-ketoglutarate. We've covered the most important part of this. The overall number of carbons can help you think about it. Now, I'm going to look at some of the steps here, but this is really more for your information. You won't hurt my feelings if you step out of here. You won't hurt them much anyways. But I just want to point out, if we look at the reactions themselves, give you some context. So we have an activated coenzyme A that could be provided from pyruvate from glycolysis, or it could come from fatty acid oxidation, or it could be made from pyruvate being made from alanine. And again, this is a very high energy thiol ester bond. You don't need to know this mechanism because it's not a simple nucleophilic attack step. This is kind of weird that this high energy bond can actually form a carban ion and nucleophilically attack the ketone. The electrons resonate up, and you leave this behind. The oxygen picks up the proton, and you now have citrate. Aconitase, I don't like the name. It's named after, I think, some plant, conate, whatever. But it really, I wish they would have called it citrate isomerase, something like that. But the point is that it's fairly simple to just reverse the chirality in this compound, which makes it okay for the next step. I see this as one of the least important steps in the cycle. But again, you should know it is an isomerase and there's no energy change, in which case that reaction can go in either direction. Isocitrate dehydrogenase, again, is one directional reaction. It drives the formation of NADH. That goes up in energy. Carbon dioxide goes down in energy. So when you're saying, if you're trying to make sense of this oxidation versus reduction, you have to say, oh, we have to not just look at alpha isocitrate and alpha ketoglutarate. We have to look at the fact that alpha ketoglutarate plus carbon dioxide will give you the total number of oxygen bonds. So here we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 carbon to oxygen bonds. I'd say isocitrate's already pretty heavily oxidized, right? There are 10 bonds here, I guess. Over here we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So this actually picked up 2 bonds to oxygen. It's a big negative energy step, and it's a regulatory step. With all of these, if you can't tell what is high energy, what is low, please do contact me. Email me, phone me, Zoom, have a Zoom meeting or something. And so in this step, we go from 6 carbons to 5 carbons plus CO2. In the very next step, we go from 5 carbon atoms to 4 carbon atom plus CO2 again. And it is this oxidation of carbon atoms that ultimately came from glucose or from a lipid that we oxidize them all the way to CO2. Now, can carbon dioxide, can that be any further oxidized? Oh, wow, maybe everybody left. Better check here.
Yes, it can.
Carbon dioxide, so how many bonds can a carbon atom have?
4?
Yeah, 4. The octet rule on that, second row of the periodic table, it can have 4 bonds. And in CO2, these are all both double bonds, right?
Oh, so it can.
And by the way, it's really in your interest to try to answer these, because my bet is that you have will remember this much better than anyone who didn't answer it. So, but yeah, that's the whole point, is carbon dioxide, you cannot get any more oxidized. It's as if in the campfire, when you burn things, you don't always have complete oxidation. You still get a lot of heat and light from it. But in the human body, you can completely oxidize that all the way down to CO2. Now, interestingly, what's really neat about succinyl coenzyme A, we're going to look at how that's used in heme biosynthesis in hematology. It isn't just that the coenzyme A can stick a acetyl group all over the place and pass it around, but it can also pass a succinyl group around, or a malonyl acid group, or there's all sorts of groups which when you link them to coenzyme A, this can bind to an enzyme and it can stick its activated group ready to form a bond all over the place. And in the citric acid cycle, it simply gets oxidized to catalyze the next step. But it's real important to be thinking that coenzyme A, hugely important in terms of acetyl groups, but it also is linked to many other groups, even a fatty acid, for example. Now, we already looked at this. The idea is that pyruvate dehydrogenase grabs pyruvate and NADH, and it passes an acetyl group over to lipoic acid, which is part of the green complex dihydrolipoyl trans-acetylase. It's going to transfer an acetyl group. And I like to represent the dihydrolipoic acid with these 2 fingers being the 2 sulfhydryls. So first, pyruvate dehydrogenase grabs the pyruvate, takes its acetyl group, and passes it to complex 2, enzyme 2, which then brings that acetyl group over to itself to catalyze the transfer of the acetyl group to coenzyme A. And that's how the acetyl group gets on coenzyme A. Then, in the meantime, the dihydrolipoyl has been reduced to 2 sulfhydryls, which come over to complex 3 and pass those sulfhydryls over to FAD to make FADH.
I had a question regarding the dehydrogenase complex. So on the prep video, you mentioned that, well, on the slide, it said that FAD is produced, but within the recording, you said NAD is produced. So,
yeah. So this is a little bit complicated, okay? So I'm glad you asked. FADH2 is made by it, but it then transfers those electrons to NAD to make NADH. And I don't like to talk about that because FADH2 is not as high in energy is NADH. So how can you pass the electrons? Well, it turns out that if you take any of these coenzymes and put them on in the context of a protein, you can actually, there's a distortion there and all. And so these electrons, which are passed to FAD to make FADH2, in the context of that enzyme, they're actually passed to a higher energy level. So it's a different type of FADH2 in the complex. And so they go there, and then that will actually pass them to the NADH. So it goes through FAD to get to NADH2.
Thank you for clarifying.
But yeah, I hate to make it that complicated, but that's actually what happens. And so in some, normally I always like to think NADH is highest energy electrons, FADH is lower, ubiquinol is lower, cytochrome C is lower, but there are contexts where electrons on a flavin ring can actually be higher than on a nicotinamide ring. I already think I'm too deep in the weeds there, but that is a really good question. And again, I don't want you focused on these, but you can see that succinate dehydrogenase, if you interpret it, takes hydrogens away from succinate. So where do those electrons come from? The hydrogen atoms come from to form FAD to FADH2. If you look at succinate and fumarate.
It came from the carbons?
Yeah, these carbons here, the carbon atoms here, each had 2 hydrogen atoms attached. When you formed the double bond, that left a single hydrogen on this one and a single hydrogen here. And so the electrons, those electrons with their hydrogen went from FAD to FADH2. And I want to mention here that going from a single carbon to carbon bond to a double bond is an oxidation, and you can drive high energy hydrogen atoms off. But it is not as high in energy as forming a carbon to oxygen bond, like where you drive a CO2 off. When you take a carboxylic acid and form a carbon to oxygen bond, that plummets in energy. That's what gives you the power to make NADH rather than FADH2. But in this case, this is such a subtle oxidation that you can form FADH2, but that's as best as you get from it. Again, we're going deeper into the weeds than most of you need, but I just want to show you that the enzyme name makes sense. And I would never say to you, the succinate dehydrogenase make a high energy NADH or FADH2. That's way too detailed. When you see dehydrogenase, think, okay, probably NADH, maybe FADH. I'm trying to give you a level of the ideas that I'm thinking about. And then fumarase simply converts the enzyme to malate, and malate goes back to oxaloacetate. And I'm out of time. I wanted to go through the citric acid cycle. The most important thing is to understand the most prevalent enzyme, what we're making, and how it's regulated to some extent. And again, you have 10 minutes before your next presentation. So I'm going to let anyone can quit. And again, as usual, if any of you have questions, I'm open for questions right now, or I have office hours this afternoon too.
Dr. Hansen, I have a quick question for you.
Yes.
So when we're talking about regulation of pyruvate dehydrogenase and carboxylase, is that solely dependent on the number of intermediates, or is there a stronger regulatory step?
The number of intermediates for pyruvate dehydrogenase?
Versus carboxylase, meaning like if we had low amounts of oxaloacetate, is that the regulatory step that would further drive carboxylase over dehydrogenase?
The level of acetyl coenzyme A absolutely will affect regulation by those. I believe that ATP, ADP is going to play a role too. And again, I'm not going to say to you, does ADP regulate, if ADP regulates several enzymes, I won't say which one does it specifically. It's going to be what is the change in, if you can interpret the change in energy, you know more than enough. Does that help answer your question? So for example, acetyl coenzyme A, will that tend to upregulate or downregulate pyruvate carboxylase?
Having high levels of acetyl-CoA would
if you have
probably,
a lot of acetyl,
yeah, I think that would down or upregulate carboxylase because we need more
wait a minute,
oxaloacetate.
that was a trick answer, man. Down
Oh
or
no.
up? No, well, you meant, I know what you mean. What did you mean? You meant,
I think you cut out there. What was your question?
I said, I know what you mean, but will you tell me again? Just was too tricky to say down or up.
Oh yeah, absolutely. So if we have a buildup of acetyl-CoA, I think that would stimulate pyruvate carboxylase because we need oxaloacetate.
Yes, that's exactly right. And I'm not going to say, you see, if you understand the logic of why you would drive one way or another, I'm not so worried about these. Is that AMP or ADP that affects that? I don't care. You know, so long as you understand energetically why it's happening, that's the big algorithm to understand. Because it'd be foolish to look at every one of these allosteric sites and enzymes and say, well, that'll, and try to memorize which particular high or low energy compound affects it.
Awesome. That answers my question. Thank you.
And I'm really hoping everyone thinks that way, because there's no way you can memorize all this stuff, right? I couldn't memorize it and I wouldn't want to. Any other questions? And again, you're all welcome to prepare for that next talk. I don't even know who goes next, but you're welcome to prepare for that. I'm just hanging out a minute in case anyone has a question, and you're more than welcome to get back with me.
Thanks, Dr. Hansen.
Okay. You all have a good day. And if you have questions, call me. Don't get bogged down in the details of every enzyme, but think about, can I simply interpret the name and make it, and does it make sense? And how is it regulated?
Thank you.
Thank you.